have these two grades there. Let me know if I make any mistake. Okay, so today we are going to be finishing our discussion on nitrogen assimilation. And so you know that the day before an exam, you can get bonus points if you answer things together. So let's wait and see if Justin or Sam get here, then maybe the three of you can work with it. I don't know, Madison, who do you usually work with it now?
I would say he over
So
there.
do you prefer to maybe work with them today?
I'm good with that, I guess. Sure.
Okay, good. So let's see how this goes.
I'll just chill here until we get to
Okay,
there.
one of the things is we started talking about assimilation. Nitrogen, assimilation of ammonium. So first we said, nitrogen assimilation is when plants take nitrate from the soil. And then in that process of taking nitrate from the soil, and then, you produce ammonium. And then ammonium assimilation is when you have ammonium and you actually incorporate it into molecules, like amino acids. So let's start talking first about nitrate assimilation. We talked about this, but no. So first of all, keep in mind this. This is going to be super important. Anytime we're talking reducing something, we're going to be using energy, some kind of energy as ATP, as NADH, as high energy electrons, whatever that is. And then when we're going from most reduced to more oxidized, then there's no energy input. We're actually doing this spontaneously and releasing some energy in the process. So this is where we left off last class. We talked about nitrate assimilation, and then we talked about nitrate reductase, so two enzymes. We're going from the most oxidized form of nitrogen to the most reduced form of nitrogen. So there's two big reductase complexes. We're reducing it in two steps. One is nitrate reductase. I don't need you to remember any of these formulas. I don't care that you remember it. Just we're reducing, and if you remember NADH or NADPH is a high energy molecule. So clearly you're using a lot of energy to reduce nitrate into nitrite. Right? How is it that you say this?
Nitrite.
Nitrate, nitrite. If I don't say it correctly, you'll know what I'm talking about. So nitrate reductase, so a lot of electrons here. And then this gives us what we have is nitrite, or this other one. And then you have to take a second step to go from here into ammonium by a different complex. And instead of NADH, you're using just something that I don't want you to really spend a lot of time right now, but next time when we talk about photosynthesis in the third kind of module, we're going to see this a lot. And it is it's a high energy compound that is also reduced. So you're reducing it with high energy compounds, reducing it with high energy compounds to go from the most oxidized form of nitrogen to the most reduced form of nitrogen. So a lot of energy is spent here. This occurs in the cytoplasm, this occurs in the chloroplast. Okay, so it's that process. Okay, this is where we start. And so let's just to keep things simple. Sorry, Madison, I'm switching you again. So you are Gonzalo.
Okay.
Where the day before the exam, every time we have questions, if you fully answer it in your groups, you're going to have up to 2 points, bonus points for Wednesday's exam. Okay, so true or false, and this one clearly cannot be just true or false. You have to give me something more. Unlike carbon fixation, that is photosynthesis, nitrogen assimilation is not affected by the presence or absence of light. True or false, and while you don't have to give me details, you need to provide me some context. And who wants to go first?
So the true, that's not just affected by the presence of light. because it's like affected by the uptake of nitrogen by microorganisms, bacteria, that then like die in the soil and then like release those like fixed compounds like ammonium and nitrate into the soils.
So you're saying this is true or this is false?
That's it's true.
Okay, it's false. But one thing I have to mention is you're not wrong in the sense that we're not exploring yet nitrogen assimilation. So, like if we're talking about nitrogen assimilation, like nitrogen from living organisms, okay? That is like nitrogen fixing bacteria, that's a different story.
Okay.
So we're gonna get there.
Cool.
So, who wants to say why this is false?
Good job. Good job.
Okay, so I don't know. Does anyone know who raised their hands first? Oh my God, it is. I don't know. Who raised their hands
And see.
That's,
Rock, paper, scissors.
heck yeah.
But no, because there's three groups.
Oh.
Yeah, you can do that.
Just versus.
Yeah. So yeah.
Good to try all three together.
So.
Maybe if someone's going to throw down paper and there's going to be two rocks.
So,
It could be two people throw down rocks and one person throws down paper.
Okay, so you're sure. Okay, good. Okay, so you go. Sure. I mean, that's why we're here today, right? Okay, so.
Is it because the nitrate reductase is in chloroplast that uses NADPH, which is affected by the presence of nitrogen?
Yeah. So you went into more detail than what I was going to, but yes. So you basically need the product of the light reactions to be able to have nitrogen assimilation. It cannot just be separate. You need the products from the light reactions, like NADPH, things that are happening at this right moment, to be able to activate the enzymes involved in nitrogen assimilation. Okay. So how is this regulated in plants? So this is what happens. There are two steps. Remember we talked about the first is nitrate into nitrite. Yeah, I'm saying that right. And then to ammonium. So this first step, that is nitrate reductase, is directly activated by light. So what happens? Darkness inactivates the enzyme and light activates it. How does this happen? Do you know what kinases are? Does anyone not know what kinases are? If you don't know, I'll just say it. Everybody knows what it is? Kinases phosphorylate products. Kinases give you phosphorylate specific amino acids, so they give you phosphate. Phosphatases do the opposite. They remove those phosphates from amino acids. And this is a very common on and off switch that all organisms have. You add a phosphate, usually you activate. You remove the phosphate, usually you inactivate. But plants don't make it easy for you, so we are going to do the opposite. In the dark, kinases are activated and they phosphorylate nitrate reductase. And when that's phosphorylated, it's gone, it's off. Only when light is present, and actually carbohydrates being synthesized, so this is all photosynthesis. So in the presence of light, a phosphatase is activated instead. That removes phosphates, and now your nitrate reductase can be activated, and you go from NO3 to NO3. Okay. Only in the presence of light. You can do that first part. But we're only halfway. We said nitrite is toxic, so it has to be quickly converted into ammonia. So what happens, there's a second part that is important. And we will revisit this very complex graph when we talk about photosynthesis. But what I need you, or what I want you to remember is, at the end of photosynthesis, so photosynthesis, the light reactions is like an electron transport chain. That's just what it is. Light energizes electrons that jump between proteins, and those high energy electrons do something, just like an electron transport chain. A little bit different. One of the things that gains those high energy electrons is called frtyguijko. The name is not important. A high energy compound that is reduced during the light reaction is required to reduce nitrite to ammonia. So unless light is going through and electrons are going through and the light reactions are happening, you cannot go from here. So light is required, one, to activate nitrate reductase, and second, to reduce nitrite to ammonia. So you need light. You need photosynthesis to be happening to do this. Okay? So, summary of this part. Lighter carbohydrates induce the phosphatase, which activates nitrate reductase, which is this first that reduces nitrate. A strong reducing agent from the light reactions of photosynthesis is required to take these last steps here. Okay? You take anything from this first part, this is the slide that you should be able to explain. Okay? I don't need you to give names, I don't need you to give NADPH or ferredoxin or any of that, but have this clear that both carbon and nitrogen fixation are linked. And then we will revisit these next time when we're talking about photosynthesis. Are there any questions about this? Okay, so now we're going to be talking from the second part. We already have ammonium, right? From here. Now, what are we going to do with the ammonium? So it's ammonium assimilation. How does this ammonium get incorporated into amino acids? And how does it go from everywhere else? So let's just review this first part. We have nitrate that is taken by plants. Remember, nitrate reductase is in the cytoplasm, so now you have nitrite, and then nitrite to ammonium is in the chloroplast. So why is that important? Your ammonium is in the chloroplast. So it makes sense because the NADPH and all of those products from the light reactions are in the chloroplast, so it makes sense your ammonium is in the chloroplast. What are you going to do with it? You're going to incorporate it into amino acids and distribute it. So I'm going to throw at you like 6 or 7 different names. I'm going to give you the names because I'm not just going to call them colors or numbers because they have actual names. But again, for me, the focus is not that you remember the names. I'll give you the names. I'll try to simplify it as much as we can. So when we think about nitrogen assimilation and distribution through the plant, I want you to think there's amino acids. And so there is a very specific amino acid. You know, an amino acid has a carboxyl group, an amino group, and then a side chain. But the regular amino acid has its amino, its carboxyl group, and sometimes carries on an additional amino group. That's what it does. So I'm a full amino acid, I'm going to load on another amino group so I can share it with others. That's how nitrogen is distributed in plants. So this regular amino acid carries another amino and then just spreads joy. So you need to name at least 2, either enzymes or molecules. Yes.
Glutamine synthase and glutamate synthase.
Yes, so glutamine synthase and glutamate synthase. Those are 2 enzymes, okay? So, that are important. So, one of your key molecules, the amino acid that is key is glutamate. Okay, so glutamate is like kind of like nucleic acid, but in glutamate. It's just iodine. So glutamate, you know, is the regular amino acid with an alpha carbon, carboxyl amino. So it's the regular amino acid. When glutamate carries an additional amino to share it with other components, becomes glutamate. Okay. So you can see this one here, and glutamate, of course, just this extra load. This one. So glutamate is the amino acid, glutamine is the amino acid with two amino. So it has another amino in the side. And basically what Lucas mentioned, so if we have glutamate and glutamine, so glutamate, that is the amino acid, glutamate synthesizes glutamate, and glutamine synthetase. Why don't they just call it synthase? You want to call it synthase. So glutamate synthesizes glutamate, and glutamine synthase synthesizes glutamine, and it's the amino acid plus that. Okay. Before we move on, then, not the point. What is essential amino acids? When I say essential amino acids, what can you think? Yes.
So amino acid that produce carbon.
Yeah, so it's amino acids that we cannot synthesize, that we need to obtain from food. Well, plants don't have that problem. You see, glutamate and glutamine, our two friends in here, you produce this, and you get everything. So, I'm not, of course, telling you that you have to learn any of these. But basically, glutamate is linked to the Krebs cycle. Once you're linked to the Krebs cycle, you're linked to the carbon cycle. So, you have these two molecules, and you can basically become every other amino acid. But the key is, the amino group will only become incorporated into glutamate and glutamine. These are your two main players. Okay. So again, just glutamate, again, is your regular amino acid. With glutamine synthase, carries the additional one, becomes glutamate. And now, this is where the new term enters. There's a third molecule that is, again, names are not important for this one, but it's a molecule that is almost like glutamate, but doesn't even have a single carbon. A single nitrogen. Okay. So you have this player here that has no nitrogen. What happens when you combine a molecule with two nitrogens and a molecule with no nitrogen? Two molecules with a single nitrogen. Okay? That's what happens. Now, a regular amino acid that now has one and two nitrogens here. And then you combine it with a molecule with no nitrogen and gives you this plus this, gives you two glutames. And then that's just basically how the cycle works. No nitrogen, then you have glutamates, you combine it, and then glutamate can donate its nitrogen. Okay. So there are two enzymes here. Glutamine synthase gives you glutamine, and then glutamate synthase. The short version, it just has this very, that gives you glutamine. And so from there, I told you, you can just basically produce chemicals. Okay. So, how do you link nitrogen and carbon metabolism? So let's try to understand this here. You have your glutamine, again, that is your regular amino acid. Glutamine synthase, now you have glutamine. Okay? Glutamine and this thing that didn't have any nitrogen because it came from the Krebs cycle. One and this and two of these give you two glutamines. And you see basically from here you just produce any carbon molecule that also contains nitrogen because you're linking both. So I don't need you to remember the two oxoglutarine. But hopefully you can remember glutamine and glutamine because those are amino acids that you can tell. Now, I don't need you to remember the name of the enzymes. You can tell me glutamate is converted to glutamine by another enzyme. That's all I care that you remember. Now, so this is the main goal. So there is an alternative way with just one enzyme. You can actually assimilate ammonium with a single enzyme. With glutamine synthase and glutamate synthase. Either name of enzyme or what happens or how is it different. Yes.
Does it use glutamate dehydrogenase instead?
Yes, so glutamate dehydrogenase. So there is an alternative way in which is again it's the same players, it's the same people doing their thing, but you just can go from glutamate to the one that didn't have nitrogen. So basically what you're having is these two exchanging nitrogen. But so here is what happens. This is an alternative way. And what scientists have found is that, I'm going to show you a study in a minute, is that this is actually a secondary, it's like a backup way that is only utilized under certain circumstances when the plant really is short for carbon in this sense. The main goal of this is nitrogen assimilation. But if the plant is low on carbon, sometimes we'll activate this as a backup. It's only one. So more than 95% of the ammonium is assimilated by the glutamate synthase glutamate. Glutamate synthase glutamate synthase. So the one with the two enzymes that we talked about. Okay. This is actually I think it's a nice study. So we're talking about this alternative pathway, glutamate dehydrogenase. So this is Arabidopsis if you cannot recognize it, this is the model organism for plants. And so what happens here is plants, or at least Arabidopsis, has three isoforms, which are three different versions of that gene. So 1, 2, and 3. So the first thing is they find that Arabidopsis usually uses glutamate dehydrogenase. Glutamate dehydrogenase 1, 2, and 3. Okay? And so the first thing they create whenever you want to figure out what an enzyme does is usually delete it and then see what happens. So what scientists did is they created triple mutants. So these 1 to 3 means you're measuring expression or transcripts. So they create a plant that doesn't have 1 or 2 or 3, has no glutamate dehydrogenase whatsoever. So first the plants can actually recover plants, so they know it's not essential. A plant can survive without any of the versions of these enzymes. Like, okay, it's not essential, the plant can live. And so here you have the regular plant, the wild type, and here's the mutant. It doesn't look the same, but it doesn't look there. Like, I mean, clearly there's a plant there. And so then what they figure out is that when they grow both of them in normal conditions, they both can thrive. They don't really see much difference. What is this telling you? That basically, this one, a plant that cannot do this whatsoever, can still live fine. But then, when they put it in under carbon limiting conditions, basically they give low light and they give even lower amounts of CO2. Then this plant cannot really thrive. It really there's just like they measure it. You can't really see here, but they measure it and there's really significant differences. So what they know is that this alternative pathway is not required for plants. They can live without it. And it's usually used when the plant is starving for carbon. So it's more a way in which you can see it here. It's a way in which basically you are obtaining more of this molecule so that you can use it in the Krebs cycle rather than in nitrogen assimilation. So while glutamate and glutamine are the main players, and this one is just like the one that eventually becomes glutamate. Whenever the plant is starved on carbon, the plant will use that alternative pathway just to have carbon. So back to what we were talking about. So we have three enzymes. The main pathway, the one that produces glutamate and glutamine, and then there's glutamate dehydrogenase, which is like glutamate and is usually under carbon. So glutamate, okay. Before we move on, I want you to make sure you can describe this, and I'm saying in your own words, which can be difficult to describe. But then again, in your own words, as in, don't worry too much about the backup, just know like there's a backup plan for the plant. But for example, if I ask you briefly describe how plants incorporate ammonium into proteins, you can give me, well, there's 3 molecules, 1 molecule has no nitrogen, an enzyme adds this, and then that's this, and this is how it gets reached. That's what I want you to be able to. No names, I forget the names of the friends, that you, if at some point in your life someone asks you about nitrogen assimilation, or if you read something, I don't know, I have one of my students that took the class the first time is actually getting his PhD in plant breeding, I think at this moment. So if you go into any of your PhD class, this will probably become useful. You know just basically how glutamate and glutamine are important players in moving nitrogen around. Okay. Okay, and so the last part is, we're talking about the nitrogen cycle. And this class wouldn't serve its purpose if we didn't talk about more sustainable ways of obtaining nitrogen plants. I was reading something yesterday that the price of nitrogen fertilizers or fertilizers in general is increasing even more, and it's increasing because of the war in Ukraine, because of all the, if we're seeing increase in gas prices, that means there's gas, and gas is going up everywhere. And we saw that incorporation of nitrogen in the field is 50% of the cost of agriculture. So fertilizers are increasing, so they are expecting the food prices to continue to increase because fertilizer prices are increasing a lot. So how do you basically put nitrogen into the plants? So we said that the main reservoir of nitrogen is atmospheric, it's in the air. So one of the limitations is how do you get that nitrogen in the air to be accessible to the plants? We briefly saw this industrialized process in which a lot of pressure and a lot of high temperatures, that N2 will become ammonium. But there are organisms that can actually do that, and they would be nitrogen fixing organisms. So the first thing that I want you to remember about nitrogen fixing organisms, you can have two types. You can have symbiotic and you can have free. Also within this same kind of example, you have nitrogen fixers that need the plant to do their thing, and some other ones that can just live on their own. And they don't really need the plant to start doing their thing. They can just usually fix that. So one of the most important groups of organisms are bacteria, or a group of bacteria known as rhizobacteria. If you ever pull a root plant and it looks like this, you will have these little balls. Do you know what these balls are for? Nodules, right? So, and this is very specific for rhizobacteria and lichens. So this is one of the things. This is why if you ever hear about crop rotation, or, like small farmers, they will tell you, well, plant your season of something and then rotate it with some kind of legume. Why? Because those are the ones that can more easily associate with rhizobia and then fix nitrogen and actually leave some nitrogen in the soil. Why? Because if you harvest beans and then you take your beans, the roots are left in the soil. Once these decompose, there's a lot of nitrogen left there in the soil. So this is very specific for legumes and rice. Yes.
I think they are not. I remember reading they're not. I think it's just because they're on a plant, it's associated with instead of the industry as ammonia. But I don't believe that they are necessary. I
Yes.
can't remember the necessary reason. I can't remember the reason.
Well, but you said no, and I already gave you the point, so you better stick with no.
It's definitely no.
Yes. Okay. So they can live without the plants in the soil, and so do legumes. You can have a perfectly fine green plant growing without rhizome. Like it's just they pair up, everybody's happier than if they don't. Yes.
Pretty sure it's because the oxygen is highly reactive, so if you have it in the same area you have nitrogen fixation, it'll actually take over and it will interact with the nitrogen.
Yes. Why will it take over?
Will outcompete any hydrogen bonds. So it's more electrophilic. So it's going to take over. It'll bond with the nitrogen over the hydrogen.
Yes. So remember, what we are trying to do is we're trying to take this N2 and reduce it to ammonia. Right? We're trying to do this. So guess who is a great electron acceptor? Oxygen. So if you have oxygen in here, what you're gonna end up reducing is oxygen, and that's not the goal. What you want to reduce is the nitrogen. You don't want it to outcompete with basically with nitrogen. So oxygen has to be away, outside from the places where nitrogen is being fixed. So if you look at this, the rate, kind of summary of the equation for fixing atmospheric nitrogen into ammonia, there's a lot of ATP involved. There's a lot of energy basically to reduce this. So yes or no? You need something more than a yes or no. What? Easy. Anybody else? So we need oxygen to be away from or not in the area. Does that mean that nitrogen fixation is only limited to anaerobic organisms? I think you raised your hand first.
Yeah. So I'm pretty sure the answer would be no, and that's because you could compartmentalize where you have the reactions at, but a lot of the organisms who may do that process anaerobically will also need to do anaerobic respiration to get the ATP required to continue the reaction to fix the nitrogen. So a lot of them would still also have aerobic processes as well, just in a different area of the cell.
Yes, so basically what happens is anaerobic or aerobic organisms can compartmentalize, just like said. So you have in some parts aerobic organisms doing your aerobic thing, and in other areas you have nitrogen fixing conditions. So what happens is many of them, even the ones that are aerobic, adapt to, okay, let's survive under low oxygen because it's just easier that we get enough oxygen, just enough that we can survive. But so what they do is they surround themselves. We're gonna see some of the parts here with nodules in areas where oxygen is just available enough for them to survive. Also, we're going to talk about this specifically. It's going to be cyanobacteria. So cyanobacteria are very important type of organisms, as you're going to see in another slide. That they do photosynthesis and they do nitrogen fixation. And the traditional photosynthesis is going to use some oxygen or require oxygen. But when you split water in photosynthesis, you have electrons, hydrogens, and oxygen. So oxygen can be a waste product. You want to do something with that oxygen. So divide photosynthesis in some cells, nitrogen fixation in other cells. And then there are some organisms that if they can fix nitrogen, they will only do it when they are under anaerobic conditions. But traditional cyanobacteria can look something like this. So you have this organism here, and then you have the regular cells doing their own thing. And they can develop a specialized compartment that they call a heterocyst. And it is like an individual cell with double cell walls and just stronger cell walls. And it is like, okay, in this room, I am going to do nitrogen fixation. And I'm going to keep all of my nitrogen fixing enzymes in this room. And there's no oxygen entering there. And then the rest of the organism can do its thing. And actually, nitrogen fixing cyanobacteria are the most important nitrogen fixation mechanism in rice fields. When you think about rice fields, I don't know if you've ever seen rice field live, but for a period of time it's floated. It's not very efficient. How do you keep dumping fertilizers? It's just not very efficient. So cyanobacteria are the way to go. And so we are going to be there in the soil. That's why many rice fields are going to be kind of grayish at the same time, rich in cyanobacteria providing the nitrogen. And so cyanobacteria in that case will be so one of the interesting things is you ever have the opportunity to have legume and a microscope at a hand. And you open one of those nodules, it looks like this. Okay? So this is how you know. If you are walking around a field and someone asks you, I think this is a nodule, how do you know if it's an active nodule? If there's actually nitrogen fixation happening in that nodule. It's a very easy test. You grab the nodule, you cut it in half like a tiny, tiny ball. You look at it inside, and if it's pink, it's fixing nitrogen. If it's not pink, it's not fixing nitrogen. And that is because this nitrogen fixation mechanism is very complex. I'm not going to get into details, but it's transported by a protein that is called like hemoglobin, which is kind of like hemoglobin, but they just legume hemoglobin. I guess someone figured that's how they're going to call it. So it's for nitrogen actually, but that's why it's good. And so what happens is, how does this get activated? How does this process even start? We said the plant can survive alone, and the bacteria, rhizobacteria in this case, can survive alone. So under low nitrogen conditions, the plant starts expressing something called nodulin genes. And the bacteria, in response to some of those chemicals, expresses something called nod genes. Not very creative, they call it the same way, but that's basically what happens. So nodulin genes by the plant and nod genes by the bacteria. And so basically this is what happens. The plant is doing fine. If it's under low nitrogen conditions, it will start expressing some of these nodulin genes, and that will release some chemicals. Is everybody familiar with what chemotaxis is? No? So chemotaxis is the process by which things are attracted, respond to chemicals. Okay? So in this case, the bacteria are attracted to the plant. And more than attracted, and they respond also to certain environmental conditions, in this case, released by the plant. So these chemicals are perceived, and then bacteria responds to that. How does it respond to that? It starts synthesizing, transcribing node factors. The node genes produce node factors. And so then the plant under low nitrogen conditions signals bacteria that are nearby, sort of nearby. They don't have to be, they aren't yet in the root, they are just nearby. Then the bacteria start synthesizing the node factors, and then that's when kind of the conversation starts, so that the bacteria can enter the plant. Can anybody tell me anything that is important? Two things that are important, or one thing that is very important for the bacteria to colonize, or something about the process of nodulation. Yes.
Or having issues that we provide, will have
Yes, but so that's more of the symbiosis, but more like about the formation, so the process of the nodulation. Can you tell me anything about it? Something that's specific to that, something roughly how nodulation happens.
They affect root hairs, or at least not increase inducing control.
Yes. So the signal specifically is with the root hairs, okay? So what happens is, let's say this is the surface of a root hair, membrane, and then you have your rhizobia. So what happens is, first the plant basically told the rhizobia to do something, right? So let's say the rhizobia are already releasing some of these node factors. Once these node factors are released, then the plant feels those node factors and says, okay, let's extend some of these root hairs, and we're going to use this curling mechanism, which is kind of like hugging those bacteria and incorporating and bringing them close to the plant and actually into the plant. So the bacteria have to go inside of the plant cells. So these node factors will induce curling of root hairs. And that is what your book is summarizing. All of this here, which is basically just rhizobia get nearby and the root hair is curled, so you're kind of hugging them. And then what happens is kind of what we briefly talked about in mycorrhizae, that years, probably millions of years of evolution, have made that the plant and the organism, in this case bacteria, in mycorrhizae was fungi, can communicate, and in this case actually the plant allows the bacteria to digest cell wall and digest membrane, and it's like, don't worry, you can do it just come here. How that works is still, how does the plant just let certain bacteria get in? Because they get into the plant and they just take place inside of the plant. So those nodules that you saw, that's plant tissue just swell with bacteria inside. So they are allowed to go in. And so basically what happens is what scientists know is that there's all of this signaling, of course, we're going to talk about some of the hormones later on, but something from very signaling very on the inside, kind of close to the xylem, will start signaling and have that root hair there develop. And then what happens is the bacteria penetrate and eventually reach some plant cells. And they will start forming something that's called the symbiosome. What is the symbiosome?
We just do the region, like that symbiotic relationship, like first begins with bacteria incorporated into the cells, like kind of like the start of the nodule.
Yeah, so basically the symbiosome is that region that is interesting because it's where the symbiosis is established. It's no longer only plant, it's no longer only bacteria. It's kind of like a different organelle because it's bacteria that are releasing the plant, but they are always surrounded by the plant plasma membrane. And in that moment, it's just signaling start occurring. So basically, when people go and study the symbiosome, it's interesting because it's different. They cannot tell where the plant ends and the bacteria begins. It's just all of the gene expression, everything is so different that it's no longer only bacteria, it's no longer free plant. And so, yeah, basically that's what happens. So the root hair, elongates and curls, the bacteria get in, and then the symbiosome here is formed. And basically it's in these, it's not organelles, it's these, how would I call it, more like symbiotic structures that again is no longer plant, is no longer bacteria. And then the bacteria just get in there, start doing their thing, and the exchange starts happening. The bacteria will get carbon, and the plant will get the nitrogen from the bacteria. Okay. Yes. Does anybody even have any questions about Wednesday?
Is it the same format?
Yeah, same format, but there's no pick and choose. There's only questions you have to answer all of them. There's gonna be a bonus, so I always throw in at least one bonus at the end. But yes, same format. So I can tell you there's something about learns potential, but is this thing going in or out? There's gonna be something about. Oh, we're going to start with the symptoms. Okay, so remember, 20% of your exam is going to be slides presented here. And just like the activity that we had the second class of this kind of model, where you had a minute to identify is it in older leaves, is it in younger leaves, the most, is it purple, is it curly, what type, and then the last question is going to be what kind of, once you identify the most important symptom, young and old, small or not, what kind of deficiency that is. And we're gonna start with that. That's gonna be 20 minutes, 20%. So you're gonna have 1 minute per slide to answer that. And then what we will have is I will give you like 10, I think it's 10 or 15 more seconds just of each one again for you to add, change, review, anything that you are not sure about, things like that. So that's gonna be 20%. And then the remaining of the exam, like the remaining 80% is gonna be something about nerves, potential, something about soluble. I shouldn't have finished the exam yesterday because I did for everything. Soluble and insoluble components moving through the soil. So is this soluble, is this insoluble, what it makes it soluble, what it makes it insoluble, and then how does it move, diffusion or bulk flow. Those are the other options. There's something also about make sure you know basic differences between mycorrhizae and rhizome. Just very basic. There's nothing in detail. I mean, at least one is bacteria, one is fungi. They already did it. And there's going to be also something about just kind of what I told you, kind of the summary process, summary as in your own words describe how nitrogen is. Nitrogen is assimilated. So nitrate assimilation, remember nitrate is, so the plants usually uptake nitrate and ammonia. Okay. Remember why nitrate, so nitrate, remember, requires the plant to use a lot of energy because it has to reduce its level. The plant has the option to just obtain ammonia, but remember why it doesn't just obtain and store ammonia. That's very important. I included a slide with a lot of colors in lecture Friday session. So make sure you know why ammonium that's already reduced is not just stored in. And then something about how ammonium is incorporated. So again, you don't need to know the names of all of the enzymes, but you should be able to describe. At least glutamine is glutamine. And how many enzymes and roughly what they do and how they become an amino acid. Okay? Yeah, everything else is the same. So bring your computers on Wednesday and you can say, oh, I'm sorry, I have someone